Hi, it's Ms. Vitale. This unit is on DNA. It is meant for AP Biology and corresponds with Chapter 15 in the textbook. On February 28, 1953, Watson and Crick discovered the structure of DNA. This laid a foundation for all of the most spectacular advances in biology up to today. They announced that it would be the secret of life. These two men were helped in this monumentous accomplishment by many other scientists, but a young scientist named Rosalind Franklin, working in almost total isolation, almost beat them to the punch. In 1951, Watson was a 23-year-old former radio quiz kid, supposedly an up-and-coming genius. He had just received his PhD in genetics and was going to Copenhagen to study nucleic acids. While on a trip to Naples, Italy, Watson found something that he knew if he answered it, he'd win the Nobel Prize. At a lecture by Maurice Wilkins, who worked for King's Lab in London, he was an English physicist and biochemist, he saw a slide of an X-ray diffraction pattern of a crystalline form of DNA. Wilkins said that the photos were the first step in uncovering the structure of DNA. Watson wanted to figure it out. He read Linus Pauling's papers on alpha helical structures within proteins. Watson then went to a lab in England to study DNA. It was a sister lab to the King's Lab that Maurice Wilkins worked at. In the lab in Cambridge, he met Francis Crick, a 31-year-old graduate student studying X-ray diffraction of proteins. Watson started nagging Crick to help him use this technique to solve the structures of DNA. Crick didn't want to get involved because he was friends with Wilkins, who was also trying to discover the structure of DNA. In November 1951, Watson went to hear Rosalind Franklin lecture. She was working at the same time at King's Lab, which is where Wilkins was also working. She had already worked on the structure of coals, and her work laid the foundation for carbon fiber technology used today to make strong, lightweight bikes, boats, etc. She learned and improved X-ray diffraction techniques and developed new mathematical techniques for interpreting the photos. She was now using these techniques to figure out the structures of biological molecules. At King's Lab, she was given the latest technology to figure out the structure of DNA. What she didn't know that was Wilkins was down the hall working on the same thing. Wilkins thought Franklin was just a technician, and Franklin thought Wilkins was a colleague. The head of the lab, John Randell, hired Franklin to work on the structure of DNA and didn't tell Wilkins. Because of this, Franklin and Wilkins didn't really get along. Wilkins talked about Franklin behind her back. They never got along, and their boss, Randell, never helped the situation. If they had worked together, surely they would have figured out the structure of DNA together. When Franklin began her work in 1951, the chemical makeup of DNA had been known for about 30 years. In 1928, Frederick Griffith, who was an English bacteriologist, was working with pneumococcus, that's Streptococcus pneumonia. He injected it into mice and tried to provoke immune defenses. He was working with two strains, the S or smooth strain and the R or rough strain. The smooth chain, the S strain, caused pneumonia. It had a coat of mucus, which is why it was called smooth, and that coat of mucus helped prevent it from being digested by white blood cells. The R strain didn't have a mucus coat, therefore was easily digested by white blood cells and could not cause pneumonia. He killed some of the S strain and injected it into mice. There was no pneumonia. Then he killed the S strain and mixed it with R strain, which normally by itself did not cause pneumonia, and he injected this combination. You would expect that two strains that don't cause pneumonia independently wouldn't cause it together, but in fact, the killed S strain and the R strain caused pneumonia and the mice died. He examined the dead mice's blood and found live S strain. Therefore, something from the dead S entered into the living R and transformed them into living S strain. He discovered transformation, which is the transfer of genes or DNA from one organism to another. This can change the genotype and the phenotype. In the early 1930s, Oswald Avery, Colin MacLeod, and McCarty were working at Rockefeller Institute in New York. They set out to determine what molecules cause transformation in the pneumococcus. In other words, what are genes made of? Scientists knew at this time that chromosomes were responsible for heredity because Sutton's chromosome theory of of heredity. They also knew that chromosomes were made of proteins and DNA. And they knew that DNA was a molecule made of ribose, nitrogen bases, and phosphate groups. Most scientists expected the transforming factor to be the protein because it performed so many other functions that they knew about. But the heat that killed the S strain would have denatured the proteins, therefore it could not have been the proteins in the DNA. Avery, MACLEOD, and MCCARTY worked for 10 years isolating many substances from the heat-killed S, mixing them with R and seeing if transformation occurred. In 1944, they concluded that only the DNA fraction of the cells produced transformation, and it was halted by an enzyme, DNA ACE, known to break down DNA but not protein. Most biologists believe DNA was a simple repetitive molecule and not capable of carrying information. Even after this work was completed, they still held out the belief that proteins carried heredity. So during the 1940s, another group of scientists were working with a different organism, bacteriophages. Bacteriophages are viruses that infect bacteria. In 1952, Alfred Hershey and Martha Chase designed an experiment to discover if DNA was the hereditary material in bacteriophages. Phages attack bacteria and they inject something into the bacteria, which takes over the cell and forces it to produce new viruses, which then burst out of the cell, and each one of those goes ahead and infects another cell. Hershey and Chase knew that only part of the virus entered the bacteria cell. Because the material that enters the cells takes it over, it must be the virus's genetic material. To determine what the material was, they used radioactive isotopes. Amino acids that make proteins contain sulfur, but not phosphorus. Nucleic acids have phosphorus, but no sulfur. Hershey and Chase grew bacteria in a P32 labeled phosphate or in a S35 labeled sulfate. They combined the two batches of bacteria with phages. Each batch produced new viruses. These new viruses were now labeled with the P32 or the S35. The new radioactive phages were added to non-radioactive bacteria. They waited just long enough for the virus to inject the bacteria, then they interrupted the process by dumping the mixture into a blender. This separated what was left of the phages from the bacteria. They centrifuged this mixture to separate the bacteria from the rest of the liquid. They found that the S35 stayed with the liquid and the P32 stayed with the bacteria. This showed that DNA was transferred to the bacteria while the protein remained outside. The Hershey-Chase experiment convinced most scientists that DNA is the carrier of hereditary information. Now that DNA was agreed to be the genetic material, the race to determine the exact chemical structure began. Biochemists already knew the chemical composition of DNA. It consists of 1-deoxyribose sugar, a phosphate group, and one of 4 nitrogen bases. The 4 different nitrogen bases are adenine, guanine, thymine, and cytosine. Adenine and guanine are purines. Those are larger molecules made of 2 rings. Thymine and cytosine are pyrimidines, and they are smaller molecules made of 1 ring. In 1950, Erwin Chargaff at Columbia University examined DNA from different species and from different sources within an organism. He found that the amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. Therefore, the amount of purines are equal to the amount of pyrimidines. This is called Chargaff's rule. So at this point in time, Rosalind Franklin had all of this information, but she didn't know how the nucleotides fit together to form a strand of DNA. Within 5 months of working on the DNA, she believed that DNA was probably a big helix with parallel chains. She knew that the phosphates were on the outside, and she determined the density of DNA. She also figured out the symmetry. Crick had the knowledge to appreciate the meaning of the symmetry. It implies that there were 2 chains, one running up and the other running down. Also, her density measurements narrowed the number of chains to below 4. Watson finally pursued Crick to help him attack the problem, and within days they had a model with 3 helixes. They asked Wilkins and Franklin to look at it, and Franklin immediately recognized it as being wrong. For another year, Franklin worked on the DNA, and Watson and Crick talked about it, but the head of their lab reminded them that the DNA problem belonged to King's lab. Franklin took an excellent picture of wet DNA, but continued working on dry DNA. In the fall of 1952, Pauling wrote a paper about the structure of DNA. Both Watson and Franklin read the paper. Franklin now knew enough about DNA's dimensions and began to build a model of the double helix. Watson went to see Wilkins, but he was busy, and so therefore he stopped to see Franklin. He saw her picture of the wet DNA, and he built his model from it. Watson got permission from his lab to build his model, and Crick told him to put the sugar phosphate groups on the outside, like Franklin had suggested. Crick decided to read a report Franklin submitted to King's lab. Crick took Watson's model using Franklin's measurements and changed it. Watson then figured out how the bases fit together. A's to T's and C's to G's fit CHARGAFF's rule. Once they were done, they announced to everyone they had discovered the secret of life. The combination of Franklin's work, Crick's work, and Watson's enthusiasm solved the problem. But when Watson and Crick wrote their results for the British journal Nature, Franklin was not cited. Franklin was never aware that Watson and Crick used her research. She worked on very important research, but in 5 years, at the age of 37, she died of cancer. In a few years after her death, the head of the labs, Watson, Crick, Wilkins, and Franklin made sure that Wilkins received the Nobel Prize along with Watson and Crick. In 1968, Watson wrote The Double Helix, and he gave Franklin the credit she deserved, but he also wrote that she was the rebellious and shrewish assistant to Wilkins. Nearly everyone present during the discovery of DNA structure attacked Watson's book, and it wasn't considered an accurate account. There are 4 key features that summarize the molecular structure of DNA. It is a double-stranded helix, it has uniform diameter, it is right-handed, that is, it twists to the right like threads on most screws, and it is anti-parallel, the 2 strands run in opposite directions. If we look at the structure, double helix is uniform in diameter. The A and the T is the same width as the C and the G. The strands are opposite, carbons 3 and 5. Each phosphate group links the 3 carbon of one sugar with the 5 carbon of the next sugar. Hydrogen bonds are in between adenines and thymines and glycine and cytosine. DNA replication is when DNA makes a copy of itself. And if you recall, when we talked about the cell cycle, an interphase of the cell cycle, DNA replicates in preparation for cell division. Since a base on one strand can match with only one other base, the sequence of bases on each strand implies the sequence on the complementary strand. Because DNA comes apart and each half acts as a template for the other half, each daughter cell winds up with one old and one new strand. This is called the semi-conservative model. Half of each parent molecule is present in each daughter molecule. Each new DNA molecule is half old and half new. Polymerization is the process that involves the assembly of many small molecules into a larger one, in this case assembling nucleotides to form DNA.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA. DNA polymerase III lays down new DNA. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.